Okay, ladies and gentlemen, you are all welcome to my YouTube channel. Today we are going to look at the basic concept of genetics. What is genetics? We are going to start with the genetics. A genetics is a scientific study of heredity and variation. So whenever we said genetics, first thing that will come into our mind is that the genetics, it involves the scientific study of heredity, and then variation. So what is heredity? Heredity is the transfer or is the inheritance or is a transfer of characters from parent to their offspring. So if a parent has a character from parent to their offspring, is what you call heredity. Like for example, if your parents are intelligent and you get that intelligence from your parent, so that is described as heredity. Another example of heredity is when we have maybe a black color and we inherit that black color from our parent. That is what we call heredity. If our parents are tall, we inherit that tallness from our parent. That also described as heredity. And then we have variation. What is a variation? A variation is like a differences that exist between the parent who their offspring. Like for example, if our parents are tall and you are now short, it means that you get the variation. It means that your parents are tall and you are now short. So that is a variation. Your parents are intelligent and you are dull. So that is a variation. That is the differences that exist between the parent and their offspring. So it means that when you have a particular character that is neither found on either your mother or father, so that type of character is described as a variation. So heredity, when you get a particular character or trait from your parent, is described as heredity. But when you acquire a character neither from your parent, so that type of a character is described as a variation. And usually variation are contributed. Variation or the main causes of variation, we have two main causes of variation. These two main causes of variation include mutation. It includes mutation. And then it also includes environmental factors. Environmental factors. So environmental factors and mutation are the two major causes of a variation. But as we continue with the class, today we are going to focus more on heredity, and then the variation will come after this video. So now we are going to we are going to look at some terms that are found in genetics, some terms associated with genetics, starting with a gene. What is a gene? A gene is a basic unit of genetics information. That is, genes determine the inherited characters. So generally, when a parent pass on a particular gene to their offspring, it means that that character will be expressed based on the gene. Like, for example, let me just give examples of one gene. Gene is usually found on a chromosome. So the genes are found on a chromosome, specifically in the DNA. Like generally in humans, there are about 25,000 known genes. There are about 25,000. There are about 25,000 known genes in humans, and so many genes remain unknown in humans. So each of these genes, each of the gene that is found in the DNA or on the chromosomes is responsible for producing a particular protein, is responsible for producing a particular protein, and a protein is a major unit of inheritance. Let me now give examples. Sometimes if your parent are talkative, that is they talk too much, means that there is a probability that they will give birth to a child that is also very talkative. The reason is because the talkativeness or the talking or the too much talk is also a control by a gene. So if a particular character is controlled by a gene, it means that that character can be passed on to the offspring. Like for example, the gene that is found in humans that make us talk is called FOXP2. It's called FOXP2. The gene is called FOXP2. So FOXP2 is a gene that is found in humans that make us to talk. So other organism that doesn't talk, it means that they do not have this FOXP2. So as I'm saying, the basic unit of inheritance is the gene. So genes are responsible for controlling a character or a trait. And where these genes are found, the genes are found on a chromosome. So you know that generally we have a cell. So inside a cell, we have chromosome. Inside a cell, we have the chromosomes. And these chromosomes are found together with a DNA. And of course, this is the DNA. As we can see clearly here, this is a DNA. This is a DNA. So in the DNA, there are number of genes. Like for example, this is a gene 1. This is a gene 2. So each of these genes is responsible for producing a particular protein. Each of these genes is responsible for producing a particular protein. And whenever there is a protein, then there will be a function. There will be a function. Like for example, as I said, we have 25,000 genes in human. So let's say we now take FOXP2 gene. So this FOXP2 gene from a DNA, this FOXP2 gene from the DNA will be converted into RNA, and then from the RNA, then it will now produce a protein. And of course, once the proteins is produced, it means that we will talk. So the more the amount of this protein are produced from this DNA, the more we talk. And the lesser the amount of this protein produced, or the less the quantity of this protein produced, then the less we talk. So that is the inheritance. So a gene, so if your parent is having this FOXP2 in higher amount or in high concentration, and then your parent also passed on that gene to the parent passed on the genes to the young ones or to their offspring, it means that the offspring or the child will also be talkative. So that is the inheritance. There is also a color of skin. What actually controls the color of skin is a melanin. So a melanin is also a protein, and there is a gene that is specific for that particular melanin. So it means that the more you produce the melanin in your body, it means that the more the black color you will be. So therefore, if your parent having low concentration of the melanin, it means that they will have a fair skin. They will have a fair skin. That is melanin. And the more you produce this melanin, the higher the concentration of this melanin, the black color you will have. So if your parent have small concentration of this melanin, it means that they have a fair skin. And if they pass on this low concentration of melanin to their offspring, it means that they are going to, the child or the offspring will have a low concentration of that melanin. And as a result of that, the child will also have a fair skin color. So then, so that is actually about the gene. So a gene is actually produced and it's responsible for controlling a character. It is actually, a gene is what determines the inherited characters. It's because if you have a particular gene and it produces a low concentration, then you pass that character to the offspring, then you will have that. Like for example, generally, some parents are very angry parents. They are very angry, and you know that the angry character is controlled by a hormone called adrenaline. So if you pass on those character or those genes to your offspring, then means that the child or the offspring will now have or will now be angry, will not be an angry child, because the first on the high concentration of that of that adrenaline gene. But of course, that is why sometimes it's very advisable to marry someone that have a character differ from your own, so that at least when there is actually sexual intercourse, so there may be a combination of the genes that will actually give a character that is differ from either of the parents. So maybe as a result of that, you may give birth to a child that will have a minimal concentration of the adrenaline, so that the child will not be an angry child. So that is a gene. And then the next thing is a genome. What is a genome? A genome is a collection of genetic information. A genome is a collection of genetics information. So like, for example, when we say genetics information is a DNA. So if you now sum all the DNA of a particular living organisms, so that is described as a genome. Like, for example, in humans, it has been known that the DNA is found in the nucleus, and apart from the nucleus, the DNA is also found in the mitochondria. So ladies and gentlemen, we should get this. We have DNA in the nucleus and we have DNA in the chromosome. So if you sum this DNA, you add them all together in humans or in any living organisms, if you sum all their DNA, that is described as genome. So it's a collection of genetics information, because the genetics information is the DNA. So collection, it means like summation. So if you add all the genetics information, that is all the DNA, it means that described as a genome. And then the next one is a chromosomes. A chromosomes is a storage unit of a genes. It's a storage unit of a genes. Why is it that it is described as a storage unit of a genes? Is because the DNA is stored and packaged in the DNA. So it means like a DNA is a trait-like structure. This is the chromosome. So it's a trait-like structure. And in the chromosomes, we have some component. What do we found on the chromosomes? Number one, in the chromosomes, we have protein. And then number two, apart from the protein in the chromosomes, we have what we call DNA. And the proteins that are found in the chromosomes, it can be histone protein, and then non-histone protein. So when you have histone, generally the DNA is very long and thin, but it is super coiled and packaged with the histone protein. So generally DNA is very thin, but the histone protein super coiled and package it, and then feeds into the nucleus. Because if you can take the DNA of humans and you stretch it end to end, it will give you 6 feet, and 6 feet is not a small height or length. So it's because of the packaging, filing, and the superfiling of the DNA. That is why the DNA can fit into the nucleus. But if not because of that, the DNA cannot be seen. So they cannot actually fit into the nucleus because it is larger, large, even more than 100 times the cell, thousands or million times DNA, the nucleus. In fact, the cell itself. So that is why it has to be packaged, coiled, and super coiled and fit into the nucleus. And then the next is the DNA. So a DNA is a nucleic acid that contains genetic instruction specifying the biological development of all cellular forms of life. So like for example, in humans, we have different cell types. Generally in average human being, there are about 37.2 trillion cells. But apart from that trillion cells, we have different cell types. We have different cell types like liver cells, kidney cells, brain cells, and we have like about 220 different cell types in humans. But what make one cells differ? What make this differentiation and development is actually due to DNA. Because generally during from the journey of life, we have when you have a sperm cell plus egg. So this sperm cell and the egg cell have their own DNA. They have their own chromosome. So when this sperm cell and egg cell join together, they will produce a zygote. So it means that in a zygote, you have a combination of a sperm DNA and a zygote DNA. In a zygote, you have a combination of a DNA from the sperm and a DNA from the egg cell. So generally, this egg cell is an undifferentiated cell. It's undifferentiated cell. When you said undifferentiated cell, it means that it's a cell that is not specified. It's not a specific cell. It does not actually differentiate itself. So, but the DNA, because this zygote will undergo mitosis to produce billions or millions of zygotes, produce a millions of these zygotes, and then these millions of the zygote cells will now, based on that, some DNA molecules will now be instructed to produce some particular proteins, and once that proteins are produced, then the cell will be specified. Like for example, some of the zygote cells will be instructed to produce hemoglobin. So once those zygote cells produce hemoglobin, it means that they are specified as a red blood cell. And some cells also will be instructed to produce maybe immunoglobulins, that is immune proteins. Then it means that those cells are specified as immune cells like beta cells or like T cells or B cells, B lymphocyte or T lymphocytes. And then some cells will be specified to produce neurotransmitters. Then the cells is now actually specified as a nerve cells. So the DNA is a language of life, is what instructs the cells to produce the specific proteins and then specify that particular types of cell. So DNA is very, very important. In fact, I usually call it the language of life. You are the way you are. You have the color of the skin. Whether you are intelligent because of your DNA. If you have black color skin, is because of your DNA. If you are smart, is because of your DNA. And whatever you are doing is controlled by the DNA. So that is why I consider DNA as a language of life. So then the next thing is chromosomal logical structure. So we have what we call a locus. What is a locus? A locus is actually a location of a gene, a marker in the chromosomes. That is a point, or is a region where a DNA, so where a gene are found. It means that in a chromosomes, there are so many sections, there are so many fragments, there are so many areas. So when we said a locus, it means it's a location where a gene are found. That, okay, if you want to get a particular gene, then I have to go to particular locus. So it's like an address. It's like an address of a gene. You can have different locus. You can have locus 1, locus 2, locus 3, locus 4. So there are some genes that have their specific locus. Like, for example, if you want to get a gene or a location of a gene, like let's say hemoglobin gene, then you have to know a specific locus where hemoglobin genes are found. So it's like an address of a gene. So a location of a gene or the address of a gene is what you call a locus, a chromosome. Then we have allele. Allele are variant form of a gene or a marker at a particular locus. In the same genes, usually genes appear as an alternative form. You can have A1, you can have A2. So like possible alleles of, like, for example, this is locus. This locus 1, there are possible alleles. It can be A1, A2, and we have locus 2, where a gene 2 are found, and it can also have it is possible allele. There is an alternative form of a gene. So you said an alternative form of a gene or a variant form of a gene is what you call allele. Like, for example, for the gene 1, we have B1, B2, B3. So it's what you call an allele. So then the next thing is a human genome. So all the human cells except the sex cells, they have, or we have 46 chromosomes. We have 46 chromosomes. And of course, in men, we have XX chromosomes in females, and we have XY chromosomes in males. But sperm cells have either X chromosome or Y chromosomes. So generally, all other chromosomes, all other cells contain 22 pair of, autosomal, all cells in the body, all somatic cells, they have what we call 46 chromosomes. They have 46 chromosomes. That is a diploid. But a sex cell contains haploids. They have haploid number of chromosomes. And when we say sex cells, it means sperm cell and egg cell. So they are one that contain only haploid chromosomes, not diploids. So as we said, we have 23 pairs of chromosomes, which is actually 46 chromosomes in total. So there are 2 sex chromosomes, and these 2 sex chromosomes is XY in males and XX in females. And there is also 22 pairs of chromosomes named autosomals or autosomes. They are called autosomes chromosomes. So we should know that we have sex chromosomes, which is XY and XX, and we have autosomal chromosomes. So then the next thing is genotype. So what is a genotype? At each locus, except for sex chromosomes, there are 2 genes. And these constitute the individual genotype at the locus. So a genotype is like the genetic makeup of a particular living organisms. So the genetic makeup of a particular organism is what you call a genotype. That is the genes. And this genotype is what actually control the phenotypic character of a particular individual. So for example, the expression of a genotype is termed as a phenotype. Like, for example, as we said, melanin is a gene. So when melanin is expressed, it means that the genotype is expressed. So once there is expressions of a genotype, then there will be a phenotype. What does that mean? What it means to express a genotype? It means that the gene is produced or have, it means that when gene produce a protein, it means that the genotype is expressed. So once the genotype is expressed, that is from gene to protein is an expression of a genotype. Gene to protein is an expression of a genotype. So once there is an expression of that genotype, then there will be a phenotype. So like, for example, if I produce a high concentration of melanin, and that melanin is produced in high amount from the gene, from that gene, a melanin protein, it means that I will have a black color. I will have black color, and that is phenotype. So phenotype is a physical appearance. It's a physical appearance. That's what you can see. For example, hair color, weight, or the presence or absence of a particular. It is like a sickle cell anemia. It's a phenotype. Like if you know that, okay, if you see someone having phenotype, you can easily know that, okay, if you see someone having sickle, you can say that, yes, this person is having sickle. And it's as a result of the expressions of the genotype, which is SS. So that is actually genotype. So a genotype is like a genetic makeup of a particular living organisms. And when that genetic makeup of a particular living organisms are expressed, it means that the gene is being converted into proteins, then there would be a phenotype. There will be a phenotype. So that is genotypes and phenotypes. So genotypes is a genetic makeup, and the phenotypes is the physical appearance of a particular living organisms. And we have what we call, under these phenotypes, we have genotypes, and the phenotypes, we have what we call dominant and recessive allele. So like, for example, these are the alleles. You see the first one here. If you can clearly see, you understand that here we have EB, and the chromosomes here we have XX chromosome. So you now have ABs that have a black eyes. And this one we have EB, we have EY, EB, and EW, and the chromosomes still remain XX, and still we have black eyes. And then you have also EW on X chromosomes, and you have Y. And of course, there is no any character on the Y. And that is why it is white in color. And then you have EBY. Sorry, you have EB on X, and Y also nothing. So you also have black colors, black eyes. So the black eyes is the phenotypes, and then the genotypes is the XY chromosomes, and the X carrying a dominant allele. So we should understand that it's what we call a recessive allele and a recessive and dominant allele. So what is a recessive allele? Like, for example, in this case, look at it, we have EP, E subscript B, and we have E subscript W. So you see here, there are two characters. The black, B stand for black, and W stand for white. So despite the fact that there is a black color and there is a white color, but at the end, the bees still appear to have a black eyes. So where is the white character? So the white character, it means it is a recessive allele. So when you said a recessive, it means that it's a character that is dominated by a dominant allele. And of course, dominant allele is an allele that masks, affects the white character. So it means that whenever there is a black and a white color in the beast, the black color is a dominant. So it usually dominates it masks, it suppresses, it suppresses the white character. So that is why here EB is considered as a dominant allele, while a recessive allele is the white color, because despite the fact that they are all there, but it's only the black phenotypes appear on the bees. So that is the phenotypes. That is what you can see. But of course, genetically, using the genotypes, you see that there is a black color and there is a white color. But despite the fact that all colors are there, but it's not expressed. It's not expressed on the bees. That is a genotypes. So a genotype sometimes is something that you cannot see, and you cannot actually predict it until when you undergo genetic analysis. But the phenotypes is what you can see. Like for example, in the case where we have a sickler individual, remember we have a carrier like AS. So if you see someone is AS, it means that phenotypically he is normal, because it will not show whether he has sickler or not. But using genetic testing, it will tell us whether we are AS or SS. And that is the genotypes. But the phenotypes is what we can see clearly. Like for example, the phenotypes here, it shows that in the second beast here, it has a black eyes. But in it, there is a white eyes, but it's not expressed because it is masked. It is dominated by the black color. So usually recessive allele is expressed when it cross with another recessive character. Like for example, in the case 3, you see, because there is no black color, it's only the white color, so nothing will mask it, and that is why the beach appears as the white. So that is more explanation on recessive and a dominant, as well as genotypes and phenotypes. So what is the dominant versus recessive? So a dominant allele is expressed even if it is a paired with a recessive character. So it expresses even if there is a recessive character. So it's a dominant character that expresses. And but in the case of a recessive allele is only visible when paired with another recessive allele. So you see, recessive allele is it's never expressed, never visible until when it paired with another recessive allele. Like for example, here we have brown and gray dog. So the effect of a dominant allele B are seen even if it is present with a contrasting recessive allele. Like for example, like in this case, you see we have brown. Also here, despite the fact that there is a gray, but in this case, it still appears as a brown. Phenotypically, it doesn't show that there is a gray color. But when the small letter b here meet with another or paired with another small letter b, then because recessive and recessive allele, when they paired with each other, then it usually give the phenotype different. So that is why here it is a gray color, because the recessive characters appeared same time. So the recessive matched with another recessive. That is why we have this gray color. And then the next thing is one locus of inheritance. So you see here we have like male and female. So in one, you see that we have AA, and in 2 we have, so A1 is male and 2 is female. So in the case of 1, we have the allele are capital letter AA, the allele are capital letter. So the allele is capital letter AA and the female have their allele as small letter AA. So I have 3 and 4. So you have child, like the child here, we have 3, 4, 5, and 6. So if the cross during inheritance, what will happen? You will have the following. Like for example, this is how it means. It means that we have a parent. The male have AA character, have AA, which is capital letter A here, capital letter A. And then the female have small letter A, small letter A. So when the cross like this, we have capital letter A, small letter A. That is what we have here. And then when this also cross with this, we'll have another capital letter A, small letter A. And that is what we have here. And then at the same time, when this A cross with another A, we'll have capital A, small letter A. Small letter A. And then when this one cross again, we'll have capital letter A, small letter A. So it means that from the first generation, all the child will be A, capital letter A, small letter A, capital letter A, small letter A. So at which situation or at which conditions where this is recessive? Because capital letter A is like a dominant and small letter A is like a recessive character. So at what situation will you have? What situation will you have these characters or these recessive characters to be expressed? Sometimes if you have, like for example, if you take this F1 generation, that is first village generation, that is capital letter A, small letter A, capital letter A, small letter A. So when you cross them, you will have capital letter AA, and then you now cross this and this, you will have capital letter A, small letter A. And then of course, then small letter A cross with this, you have capital letter A, small letter A. And then this small letter A cross with small letter A, then you have small letter AA. So you can have, you see here, because a small letter A cross with another small letter A, then you now have the recessive characters paired. You have the recessive characters paired. So this is what actually happened in the inheritance. So one locus, that is one locus inheritance. So you see, so when you have capital letter A, small letter A, they are called heterozygotes. So it means that they are the same allele, but one is dominant and the other one is recessive. So that is why they are heterozygote, because they are not the same. But if you have capital A is homozygote, or small a is homozygote. So if you have the same allele, it means that they are called homozygote. And if you have different allele, they are called heterozygote. So now we are going to look at the Mendel's first law. So the two members of a gene pair segregate from each other into the gamete. So that half the gamete carry one member of the pair and the other half carry the other member of the pair. So like for example, this is, we have capital Y, small y, small y, small y. So when there is this segregation of the two members of a gene pair segregate from each other into the gamete, so that half the gamete carry one member. Like for example, here we have capital Y, small y. So you see the small y is now segregated into the half. And then capital Y will also be segregated. And also in the case of, like, this is a gamete production. So also at the same time, we have small y, small y. So this small y will also segregate into small y, and also the other one segregate into small y. So here, the small y, this small y here, this small y here, it cross with one small y, and that is why we have small y, small y. And then at the same time, we have capital Y, which also segregate itself, will now cross with another small y. And that is why we have capital Y, small y. So according to this first law of inheritance, it means that if you have two members of a gene pair, so they segregate itself. Like for example, if you have capital A, small a, then the capital A will now segregate itself. The small a will also segregate itself. Capital A, small a. So the capital A will segregate itself. The small a will also segregate itself. And then maybe you have small y, and then the small y here will also segregate itself, and this one will now segregate itself. So after segregating to the half, each of the gamete, then one of the pair and the other half will carry the other members of the pair. So that is why, like for example, this one pair can cross with this, and this one can also cross with this to produce a character. And that is what happens during gamete production. So when you have a two pairs of a gene, that is what usually happens. That is the Mendel's first law of inheritance. When two pairs of a genes pair or they segregate each other, the half of the gametes, and then each of the half of the gamete will now pair with the other member to form a particular character. And this is usually happened during gamete production. So this law is actually, according to that, is considered as the first law of Mendel, and it's usually happen during gamete formation. So how do you calculate the probabilities? Calculating probabilities in genetics. So we want to predict the patterns of inheritance of a trait and disease in pedigrees. E.g., we want to know the likelihood that a dog chosen at random from the study population will have a blue eyes. Will have the blue eyes. So we have a blue eyes and we have brown eyes. And the blue eye, they are 10, and the brown eye, they are 30. So since the blue eyes are 10, so we are now going to take the probability, and we are now going to take 10 divided by 30, and will give us the probability of the blue-eyed dogs, and the brown-eyed dogs will be 20 over 30. Now remember, probability, when you take everything, it will give you, when you take everything, it will give you 1. So the summation of all probability should give you 1. If it is 0, it's not possible. It means it's not possible. And if it is greater than 1, it means that also it's not possible. So everything in probability must be between 0 to 1. So like for example now, assuming now maybe you are AA and you are married to a woman with SS or with AS, or assuming you are AS and you married a woman with SS. So we can ask you, you calculate what would be the probability of you giving birth to a baby with a sickle. So to do that is simple. You are AS and you are also married AS. So if you are AS and you married AS and you now want to calculate the probability, so you now cross A and A, you have AA, then you cross AS, you have AS, and you cross SE, you have AS again, then you cross SS, then you now have SS. So you can now calculate what is the probability, because now AA is normal, AS and AS, they are carrier, and SS, they have secular. So now the question is, how do we now calculate what is the probability of giving birth to SS child? So since we have only one SS, and when we sum everything here, the child here, there are 4. So it means that in every of 4 child, there is a chances that you will give birth to one singular child. And to do the calculation, you now say 1 over 4. 1 over 4. So you now say 1 over 4. Of course, when you take 1 over 4, it will give you 0.25. And then, what is the probability of you giving birth to a singular carrier, to a carrier? So there are 2. So you have 4, you have 2 over 4. It's now going to be 2 over 4, which is 0.5. And what is the probability of you giving birth to AE? It's 1 over 4. So 0.25. So this is how to calculate probability in genetics. How to calculate probability in genetics. So now the next thing is X-linked inheritance. X-linked inheritance. So different results obtained from reciprocal crosses between a red-eyed and a white-eyed Drosophila. So assuming now you have a red and a white-eyed Drosophila, and you have also a white and a red-eyed Drosophila. So after crossing, according to the first law of inheritance, so each of these genes bearing gametes will be segregated and then cross with the partner. So it means that this red color will segregate itself. The red color now segregate itself to now segregate itself as this. The white also will segregate itself. The white also color will segregate itself here from B, and also the red color will now segregate itself from here. So when crossing, these reds and whites will cross with each other, and then for the F1 generation, you will have, first you will have reds, you will also have another reds, and then you will also have another reds and then whites. Because after you cross, you have something like this. You have, when you cross this with this, when you cross this and this, because this red is a dominant, or is, like for example now, the gene responsible for the eye color is X-linked. It means that it's found on the X chromosome. The females have 2 X chromosomes, while males have 1 X chromosome and 1 Y chromosome. So that is why after crossing A and B here from F1 generation, all the child will have a red color. Will have a red color except one. Because at the end, since it is X-linked, this white color will now cross with another white color. It means that you have a character that will be expressed that is different from the other, because the white color here appears as a recessive. Now, of course, for the L2 generation, we are also going to have this set of child. So the next thing that we are going to look at is the second law of inheritance. The second law of inheritance. So according to the second law of inheritance, it said that different genes paired are sought independently in gamete formation. Like for example, this law is true only in some cases, so it's not applied to every cases. The genes and pairs on a separate chromosomes are sought independently at meiosis. So genes are sought on separate chromosomes are sought independently at meiosis. So the next thing is medical genetics, which we are going to look at it in the next video. So today we look at the first and second law of inheritance. and we will continue in the next video. We'll look at the genotype, medical genetics. Thank you. If this is the first time you are coming to a YouTube channel, please